In the shifting landscape of modern science, there are moments when technological innovation and biological discovery converge so tightly that they challenge long-held distinctions between what is natural and what is synthetic. One of the most fascinating developments in this realm is the DishBrain experiment, conducted by a team of researchers based in Australia and led by Cortical Labs in collaboration with multiple neuroscientists and bioengineers. The project centered around the concept of growing living neurons in a controlled laboratory setting and connecting them directly to a computer interface. These neurons were then given a task to complete, playing a simple video game, using nothing but their inherent biological ability to process signals, respond to stimuli, and adapt over time. What resulted was not only a new type of biological experiment but a redefinition of what it means to learn, to process information, and perhaps, at some distant horizon, to think. The origin of the DishBrain project can be traced to long-standing scientific interest in the behavior of neurons when removed from the body and placed in laboratory cultures. It had already been demonstrated for decades that neurons could be sustained in dishes for extended periods, and that these neurons would naturally form synaptic connections, building networks similar to those found in the brain. However, these cultures were typically used to study biochemical interactions or the effects of drugs, not to perform interactive tasks. The DishBrain experiment took this concept much further by embedding these neurons into a closed-loop system where they could receive real-time feedback from a virtual environment and adjust their behavior accordingly. The system worked by growing hundreds of thousands of neurons in a flat dish layered with microelectrodes. These electrodes served as both sensors and transmitters. On the one hand, they captured electrical signals emitted by the neurons as they fired. On the other hand, they sent electrical impulses back to the neurons in a controlled and patterned manner. This bidirectional communication formed the foundation of the experiment. The neural network in the dish was not merely being monitored, it was being trained, nudged, and responded to by a digital system. The interface created a dynamic environment in which the neurons could exhibit plasticity, the ability to rewire and adjust their connections based on input. To give the neurons a task, the researchers turned to a simplified version of the video game Pong. In this game, a ball moves back and forth across a screen, and the goal is to position a paddle so that it hits the ball and sends it back. The neurons in the dish were connected to a virtual paddle. When the paddle succeeded in hitting the ball, the neurons received a form of predictable, structured electrical feedback, which researchers interpreted as a reward signal. When the paddle missed the ball, the feedback was chaotic and less organized, designed to represent a kind of negative consequence. Over time, the neurons began to change their firing patterns in response to these outcomes. Their behavior shifted from randomness to directed responses. This indicated that the neurons were not only reacting to stimuli but were learning from experience. What made this experiment so remarkable was not that neurons could fire or respond, those facts were well known, but that they could learn to associate specific patterns of feedback with specific behaviors and then modify their activity accordingly. In short, the neurons learned to play Pong, albeit at a rudimentary level. There was no external software telling the neurons what to do. The system was not being controlled by artificial intelligence or pre-programmed algorithms. Instead, the learning emerged from the neurons themselves as they formed new synaptic connections, adjusted their excitability, and refined their collective behavior to optimize their response to stimuli. This was the first time that a synthetic neural system composed of living cells had been shown to demonstrate this kind of adaptive behavior in a closed-loop environment. From a scientific standpoint, the implications were enormous. First, the experiment demonstrated that the capacity for learning does not require the full complexity of a brain. Even a network of a few 100,000 neurons, when given the right interface and feedback structure, could demonstrate measurable learning. This challenged existing models of cognition and forced researchers to reconsider the fundamental elements required for intelligent behavior. Second, it provided a new model system for studying neurological diseases, brain development, and synaptic plasticity in a controlled environment. Rather than relying solely on animals or computer simulations, scientists could now observe and manipulate a living, learning neural system in real time. The DishBrain project also sparked significant philosophical and ethical debate. While the neurons did not possess consciousness or self-awareness, they were exhibiting behavior that many people associate with thinking or decision-making. The fact that biological material was interacting meaningfully with a digital environment raised questions about the boundaries between machine and organism. Did this hybrid entity qualify as a form of intelligence? Was it simply a complex reflex system, or was it something more? Should such a system be afforded any kind of moral consideration? These questions were not academic distractions, they were central to how the technology might be used and regulated in the future. Another layer of complexity emerged from the discussion about sentience. The researchers were careful to emphasize that the neurons in the dish brain were not sentient. They had no sensory organs, no awareness, no internal representation of the world. However, they did exhibit behavior that was informed by past experience and shaped by outcomes. This raised concerns about the future trajectory of such research. If neural networks could continue to be scaled up, become more interconnected, or be augmented with memory, would they begin to cross a threshold where subjective experience or suffering became possible? These are not easy questions, but the DishBrain Project made it clear that they would soon become necessary to confront. Technically, the experiment provided important insights into how to engineer bidirectional interfaces between organic and digital systems. The feedback loop had to be fine-tuned so that the neurons were neither overwhelmed by input nor left in a passive state. The timing of the signals, the amplitude of the electrical pulses, and the pattern of feedback all had to be carefully calibrated. In addition, maintaining the health of the neurons required a sophisticated life support system involving nutrient delivery, waste removal, temperature regulation, and sterile conditions. This biological infrastructure was as important to the success of the experiment as the digital interface itself. One of the lesser-known but equally intriguing aspects of the DishBrain research was the way it affected the culture of neuroscience and computer science. For many years, these fields had operated with largely separate methodologies and philosophies. Neuroscience focused on understanding the brain as a biological organ, while computer science developed models of computation based on logical and mathematical rules. The DishBrain experiment forced a convergence. It became impossible to talk about neural networks in purely biological or computational terms. The system was both alive and programmed, both organic and engineered. This created a new interdisciplinary space where biology, engineering, philosophy, and ethics could no longer afford to remain isolated. In terms of public perception, the DishBrain captured the imagination of many outside the scientific community. It resonated with deep cultural ideas about artificial life, machine intelligence, and the blending of human and machine. While some viewed it as a marvel of modern science, others expressed concern about the ethical trajectory of such technologies. The idea of growing neurons in a lab and teaching them to play games struck some as a dangerous step toward creating life forms that might one day demand recognition or rights. Whether those fears are grounded or speculative, the fact that they emerged at all speaks to the emotional and symbolic power of the DishBrain experiment. Although the neurons used in the experiment were derived from stem cells and cultured in strictly regulated environments, the very act of training them introduced a dimension of agency that was previously reserved for living organisms with brains and bodies. The neurons were not acting randomly, they were acting in patterns shaped by experience. This opened up a range of questions about how intelligence is defined. Is intelligence the ability to solve problems? Is it the capacity for memory? Is it the expression of intent? The DishBrain experiment did not provide definitive answers, but it reframed the questions in a way that made them more urgent and more relevant. Another important outcome of the DishBrain research was its impact on the future of brain-computer interfaces. Until this point, most such interfaces focused on extracting data from the brain for use in controlling external devices, such as robotic limbs or communication systems for people with paralysis. The DishBrain flipped this model by treating the neural tissue not as a source of signals, but as a computing unit in its own right. It demonstrated that neural tissue could be not just a subject of study, but an active participant in digital systems. This suggested new possibilities for hybrid computing systems that combine the best features of organic adaptability with the speed and structure of silicon-based technology. Looking forward, the DishBrain project set the stage for a new era of research into biological computation. It showed that learning, adaptation, and memory are not the exclusive domain of large, complex brains or advanced algorithms. They can emerge from relatively simple neural networks when those networks are placed in the right conditions and given the right feedback. It also highlighted the importance of context, showing that neurons are not merely reactive but can become functionally intelligent within interactive environments.